Welcome and thank you for attending Get to Know Vibrant Neurotransmitters Test Spotlight on Serotonin. My name is Adair Anderson. I am a clinical lab educator here at Vibrant America, and I will be your moderator this evening. I will be responding to the live chat and Q&A throughout the presentation. And following the presentation, there will be approximately 15 minutes for live Q&A. As a reminder, all attendees are in listen-only mode. So please submit your questions through the virtual Q&A or chat applications. The content of this webinar is intended for educational and informational purposes only. The information is not intended to treat, cure, or prevent any condition or disease. If you desire medical advice, diagnosis, or treatment, please consult with a medical professional or healthcare provider. And now, I have the pleasure of introducing today's presenter, Kerri Louati, who is a clinical lab educator, and she is also on the educational specialist team here at Vibrant America. Kerri Louati is a registered dietitian and certified diabetes educator with a background in integrative and functional medicine. Kerri completed her undergraduate degree in 2009 from Bastyr University, an institution rooted in naturopathic principles, and her master's degree in nutrition and functional medicine from the University of Western States. Kerri has extensive experience working in a variety of clinical settings. She specializes in gut-related disorders, neurological disorders, and G-tube feedings with an emphasis on the pediatric population. Without further ado, I'll turn it over to you, Kerri.
Thank you so much for that introduction, Adair, and welcome to the presentation, Getting to Know Neurotransmitters with a Spotlight on Serotonin. We'll begin with an introduction to neurotransmitters and the neurotransmitter test. We're going to be going over a general overview of serotonin, talking about some factors that affect serotonin levels. We're going to be discussing intervention considerations, and we're also going to review a case study with time for Q&A at the very end. So let's get started. So let's begin with an overview of neurotransmitters. Neurotransmitters are endogenous chemical messengers that are secreted by neurons in response to an electrical signal. Neurotransmitters are located in the axon terminal, which is right here, and stored within thin-walled sacs called synaptic vesicles here. Now, when a neuron receives a message or an electrical signal, the electrical charge causes the vesicles to fuse with the nerve cell membrane and release the neurotransmitters into the synaptic cleft. Each type of neurotransmitter binds to a specific receptor on the target cell. This is the postsynaptic neuron. And after binding, the neurotransmitter can then trigger a change in the action of the target cell. This can be an electrical signal in a nerve cell, a muscle contraction, or the release of hormones from a gland. So depending on the type of neurotransmitter and area of the body, it may lead to inhibition or excitation in the postsynaptic neuron. Neurotransmitters are involved in an intricate symphony of communication within the nervous system, and they're involved in many physiological processes and functions. These range from regulating mood, heart rate, sleep regulation, digestion, appetite, muscle movement, and so much more. So neurotransmitters can be classified into different categories depending on the actions that they have. Inhibitory neurotransmitters block or prevent chemical messages from being passed along any further. In other words, they inhibit those chemical messages. Serotonin, GABA, and glycine are examples of inhibitory neurotransmitters. And what happens is when an inhibitory neurotransmitter binds to that receptor on a postsynaptic neuron, it opens up potassium channels, and that allows some of the positively charged potassium to leave that postsynaptic neuron, causing the neuron to hyperpolarize and become more negative. That makes it less likely to fire. Now, on the other hand, excitatory neurotransmitters excite the neuron and cause it to fire off the message, meaning the message continues to be passed along to the next cell. Examples of excitatory neurotransmitters include dopamine, epinephrine, norepinephrine, acetylcholine, glutamate, aspartate, and histamine. This happens because the binding of an excitatory neurotransmitter to a receptor allows positively charged sodium to enter into that postsynaptic neuron, causing it to depolarize. Therefore, it makes it more positive, and it can have an excitatory effect, meaning it's more likely to fire. Some neurotransmitters, such as serotonin and dopamine, can have dual inhibitory and excitatory effects, depending on the situation and the area of the body. One of the things that's important to understand when looking at neurotransmitters is that the total inhibitory or excitatory effect on the nervous system is important. Neurotransmitters may be within the normal range, but high normal and low normal values can influence that total impact of inhibition or excitation in the nervous system. So assessing all low normal and high normal values is really helpful. These are the markers tested on the neurotransmitter test. Vibrant includes 30 different markers. The test not only includes the major neurotransmitters, but also many metabolites, precursors, and end products to allow providers to assess the entire metabolism involved with these neurotransmitters. The test includes amino acids such as tryptophan, tyrosine, taurine, glycine, serine, and aspartate. Markers along the serotonin pathway are measured. These include 5-HTP, serotonin, and 5-HIAA. Markers along the kynurenine pathway include kynurenic acid and quinolinic acid. And the catecholamine pathway looks at markers such as L-DOPA, dopamine, DOPAC, 3-MT, HVA, norepinephrine, epinephrine, normetanephrine, metanephrine, and VMA. So definitely a very extensive list of intermediates that we look at on the catecholamine pathway. The test also includes trace monoamines such as tryptamine, tyramine, and PEA, which is phenylethylamine. And other neurotransmitters that we look at include glutamate, GABA, histamine, acetylcholine, and oxytocin. Ratios of some of these neurotransmitters and metabolites are also calculated, and this provides important information on the activity of the enzymes as well as the pathway activity as well. The report will also depict diurnal rhythms for norepinephrine, epinephrine, and creatinine. So this is the first page of the report, and it summarizes all of the abnormal results. This page also contains the diurnal rhythm graphs for norepinephrine, epinephrine, and creatinine, which is not shown here. This is really helpful to understand the time of day that correlates with the levels that are measured. The next page, which begins the complete report, shows the results for all inhibitory neurotransmitters and excitatory neurotransmitters. Any abnormal value will populate a comment to give you some more information about that specific marker. The last page shows results for other neurotransmitters. It also provides results for the diurnal rhythm for norepinephrine, epinephrine, and creatinine. And lastly, it also contains the ratios that we look at as well. So this neurotransmitter test, the way that I like to describe it is that it provides a snapshot in time of your neurotransmitters and metabolites. So you're not only able to understand the neurotransmitter levels, but also the metabolism of neurotransmitters to really hone in on what the issue or reason for imbalance is. When you look at mental health disorders, for example, most of the treatment interventions, especially conventionally, involve a trial and error approach with medications. Using this test takes the guessing out of the game and allows providers to formulate interventions based on the patient's specific lab values as opposed to guessing. Along the same lines, providers can also use this test to monitor medication or supplement efficacy and to make changes to a patient's medication or supplement regime. Ordering neurotransmitters can be helpful for a variety of different patients that may be suffering from neurological disorders, mental health disorders, and a variety of other symptoms. And we're going to go into some of the symptoms that might be specific for serotonin imbalances. I'd like to go through a few helpful tips that may guide you when interpreting a neurotransmitter test. The first step in interpretation is to follow the pathways. It's important to understand the pathways of the neurotransmitter involved and to use all of the information available to determine why a neurotransmitter might be imbalanced. We'll be discussing specific serotonin pathways shortly. The next tip is to look for trends. Is there a suspected nutrient deficiency that may be impacting a particular neurotransmitter? Is there a trend where this nutrient deficiency may be impacting other neurotransmitter pathways? We also discussed already the importance of balancing inhibitory and excitatory neurotransmitters. It's typically recommended to support inhibition before excitation in the nervous system, but understanding the overall inhibitory or excitatory effect in the nervous system can be extremely helpful. Address root causes. So for example, are there neurotransmitter imbalances because there is digestive insufficiency impacting the availability of precursors? Or is there significant stress that may be shifting the neurotransmitter pathways? Focusing on the root causes is going to be really helpful. Understand the symbiosis. Neurotransmitters do not function in isolation. There's an intricate symphony of communication that also depends on the activity of other neurotransmitters and hormone levels in the body. And lastly, pair results with clinical symptoms. This is also really important because neurotransmitter levels can differ in the peripheral nervous system compared to the central nervous system. So make sure that the symptoms correlate with the interventions that you are making. So now we're going to put the spotlight on serotonin and dive a little deeper into this neurotransmitter. So let's go over some basic information about serotonin. Serotonin is commonly known as the happiness neurotransmitter. It's classified as an inhibitory monoamine neurotransmitter, and it has functions both as a neurotransmitter, but also as a hormone in the periphery. Serotonin is unable to pass through the blood-brain barrier. So serotonin that is synthesized in the periphery cannot cross into the central nervous system. However, there are transport systems in place that can move serotonin from the central nervous system into the peripheral nervous system. Therefore, the urine neurotransmitter test may potentially reflect what is happening in the central nervous system through this mechanism. Less than 5% of serotonin is produced in the raphe nuclei in the brainstem. There is a different isoform of tryptophan hydroxylase, which is the rate-limiting step in serotonin synthesis, which we'll talk about, that is predominant in the central nervous system. This is called tryptophan hydroxylase 2, abbreviated TPH2. We'll discuss the importance of this a little later on. More than 95% of serotonin is actually produced in the enterochromaffin cells in the gut, primarily by the isoform tryptophan hydroxylase 1. Peripheral cells can also produce serotonin. These include pancreatic beta cells, adipocytes, osteoclasts, and mast cells. So they will play a role as well. Platelets are a known storage site for serotonin, storing roughly 2 to 3% of total serotonin. And even though they can degrade serotonin, they do not have the ability to synthesize it. So typically, platelets levels of serotonin are a reflection of serotonin produced in the gut. Serotonin exerts multiple functions, both in the central nervous system and in the periphery. It's probably most well known for its role in mood, behavior, and depression, although it can affect almost every area and system in the body. Starting with the gut, serotonin plays a prominent role in motility, visceral hypersensitivity, nausea, vomiting, gastric emptying, pancreatic secretions, and colonic tone. Focusing on motility, we know that high levels of serotonin can increase peristalsis and lead to higher levels of motility that may be associated with symptoms such as diarrhea. On the contrary, lower levels of serotonin can often slow motility and be associated with symptoms such as constipation. Nausea and vomiting are other common symptoms that are associated with higher serotonin. And when you look at medications geared towards nausea, for example, ondansetron, typically known as Zofran, acts as a 5-HT3 antagonist. So while we typically think of neurological conditions as the prime candidates for a neurotransmitter test, any patients with GI dysfunction would benefit from taking a closer look at how neurotransmitters might be affecting gut function. There are many mood and behavioral symptoms that correlate with serotonin imbalances. These range from aggressiveness, anger, addiction, compulsive behaviors, and obsessive thoughts. So this is another area where if your patients are experiencing any of these behaviors, it might be a good idea to see if there are any serotonin abnormalities. Serotonin plays an important role in the regulation of sleep. It stimulates the part of the brain that controls sleep-wake cycle, and it's also a precursor for melatonin synthesis. Serotonin can impact sexual health and behavior. So for example, serotonin levels that are too high can cause a decrease in libido. Levels that are too low, however, may contribute to a higher than usual libido. So that might be helpful depending on what your patient's experiencing. Higher levels of serotonin can increase satiety, while lower levels may be associated with increased appetite. So you may see this with children that are don't have sufficient appetite, or in patients that are trying to accomplish weight loss. Looking at bone health, osteoporosis has been associated with higher levels of serotonin. So this is another area where we may not typically think of doing a neurotransmitter test, but it may provide valuable information for patients suffering from bone loss. Serotonin plays an important role in blood clotting, with higher levels increasing platelet aggregation. And serotonin also regulates cardiac function, particularly heart rate and vasodilation. It has an important role in pain regulation, where it's been shown to have hyperalgesic and analgesic effects. And other functions that are important include immune system function, increased inflammation with higher serotonin levels, regulation of metabolism, and other functions like respiration and motor control. This table nicely categorizes symptoms and some conditions based on the serotonin level. We already discussed some of these, but if your patient presents with any of these symptoms or conditions, it would be helpful to do a neurotransmitter test to understand what the serotonin levels are, as well as the entire metabolism cascade to understand why serotonin levels are at the level they are. Keep in mind, this is not a comprehensive list of low and high symptoms or conditions, but you can see some of the ones that we've already discussed, some very pertinent symptoms like poor gut motility and constipation, definitely some emotional and behavioral symptoms, social phobias, emotional sensitivity. You might be dealing with a patient that has dependence behaviors, addictions, cravings. Even headaches can be associated with low serotonin, anxiety as well. High serotonin has obviously different symptoms. Irritability, agitation, anxiety can also be a symptom of high serotonin, as well as migraines can also be a symptom of high serotonin. And then you might see high serotonin in certain conditions. I've listed some here, such as autism, which we'll talk about some interesting facts related to that, osteoporosis, which I already mentioned. So a patient that has any of these symptoms or conditions would definitely be a prime candidate for looking a little bit closer at those neurotransmitter levels. One thing that's important to understand when working with serotonin is that extremely high levels can result in a serious condition called Serotonin Syndrome. This condition can be life-threatening and often occurs when a combination of medications or supplements that affect serotonin are taken together. This might be seen, for example, if somebody was taking an SSRI, which is known to increase the length of serotonin activity, in combination with a supplement or medication like 5-HTP, which can increase total serotonin levels. Some of the symptoms to be mindful of include diarrhea, nausea, vomiting, agitation, shivering. These are some of the more minor or mild symptoms. And then some of the more severe symptoms include confusion, delirium, tachycardia, fever, seizures, and even death. So whenever I'm working with a patient and we're manipulating serotonin levels, especially trying to increase them, I think it's always important to educate the patient on these symptoms so that they can be empowered as well. So in July of this year, a new study was published in Molecular Psychiatry, where the researchers wanted to evaluate the evidence on whether depression is associated with lowered serotonin concentration or activity. This was a systematic umbrella review of the research, and it's important to note that they did have some exclusion criteria, including animal studies and studies exclusively concerned with depression in physical conditions like post-stroke or specific subtypes of depression, like depression in children. But the conclusion of the article was that huge research effort on the serotonin hypothesis has not produced convincing evidence of a biochemical basis to depression. We suggest it is time to acknowledge that the serotonin theory of depression is not empirically substantiated. So this is a really controversial topic, but it's important to be aware of this research. One of the takeaways and messages that I really got from reading this article, which we know far too well in functional medicine, is that it's never just one thing. It's becoming more clear that depression cannot be boiled down to a single cause, but rather a variety of factors. But this does not mean that it is not an important factor to look at. It means that it may not be the sole factor responsible for depression. So now we're going to begin to follow the pathways. This is extremely important and helpful when looking at serotonin. In order to understand serotonin, we first have to understand the metabolism of tryptophan, since it is the amino acid precursor for serotonin synthesis. Tryptophan can be metabolized down multiple pathways, including the serotonin pathway, which you can see up here in the green, the kynurenine pathway on the left in the purplish blue, and for indole metabolism on the right. The main pathway for tryptophan metabolism, which metabolizes roughly 90% of tryptophan, is the kynurenine pathway, which we're going to talk about. But for now, I wanted to show you this visual of tryptophan metabolism because this is going to be an important factor in serotonin levels, and it's really helpful when you have that visual to look at and understand. So let's take a look at the specific serotonin pathway. In order to synthesize serotonin, there must be sufficient tryptophan available. It's important to know that tryptophan does compete with other large neutral amino acids, such as branched chain amino acids and tyrosine, for transport into the brain in order to synthesize serotonin. But starting with the first step of the pathway, it begins with the conversion of tryptophan here to 5-HTP. And it requires the enzyme tryptophan hydroxylase, which is the rate-limiting enzyme. Studies have identified 2 isoforms of the tryptophan hydroxylase enzyme. I already briefly mentioned, but TPH1, which is present mainly in the peripheral organs like the gut, and TPH2, which is associated with the nervous system and present predominantly in the brainstem. This enzyme requires nutrient cofactors, including tetrahydrobioterran, which is abbreviated as BH4, iron, and oxygen. 5-HTP is then converted to serotonin by the aromatic amino acid decarboxylase, which requires vitamin B6 as a nutrient cofactor. Once serotonin is formed, it can then be used to synthesize melatonin via 2 different enzymatic reactions requiring methyl support from SAMe and acetyl-CoA. Serotonin is broken down to 5-HIAA by the MAO enzyme, which is monoamine oxidase, the subtype A, and that requires vitamin B2 as a nutrient cofactor. Because the MAOA enzyme is located intracellularly, serotonin that's secreted needs to be taken back up into the cell before it can be inactivated and broken down. Since serotonin is also a highly charged molecule, it's not able to passively diffuse across the cell membrane, and it requires transport by serotonin reuptake transporter, which I'm going to abbreviate as SERT, and we'll talk about the importance of that. So we're going to take a look at a variety of factors that can impact serotonin levels. This is not a comprehensive list, but it encompasses some of the most common and applicable information. This includes upregulation of the kynurenine pathway, nutrient deficiencies, enzyme activity and genetics really fall under the same category more or less, SERT transporter activity, medications and supplements, hormones, other neurotransmitters, and gut health. So let's begin with the kynurenine pathway. This is the dominant pathway for tryptophan metabolism. And kynurenine is produced, you can see kynurenine is the first intermediate. This is produced by the action of tryptophan 2,3-dioxygenase, which is abbreviated as TDO, which you can see right here. And TDO is induced and upregulated by glucocorticoids or excess levels of cortisol. The other enzyme involved in this first step is indoleamine 2,3-dioxygenase, which is abbreviated as IDO. IDO is influenced by certain inflammatory stimuli with interferon gamma as the most potent inducer. Infections have also been shown to upregulate this pathway. So once kynurenine is produced, which is this first intermediate, it's further metabolized along two arms of the pathway, one leading to the production of the neuroprotective kynurenic acid, which we can see here, which we do test for on the neurotransmitter test. Kynurenic acid acts as an NMDA receptor antagonist. The other arm forms the more neurotoxic quinolinic acid, which you can see here, which acts as an NMDA receptor agonist. This is also tested for on the neurotransmitter test. The effects of increased tryptophan metabolism along the kynurenine pathway can be dual in nature. The first is there's reduced availability for serotonin synthesis, and there's also increased production of neuroactive kynurenine pathway metabolites. While this is the dominant pathway for tryptophan, it's important to understand that increased levels of cortisol and inflammation can upregulate this pathway even more. Another important point about this pathway is that the end product of this pathway leads to endogenous niacin synthesis. Niacin is a precursor for NAD, which plays a role in the electron transport chain and in the synthesis of ATP. Niacin is also a precursor to NADP, which functions as a coenzyme in oxidation reduction reactions. It's possible that the factors that upregulate the kynurenine pathway may be a response for increased need for more niacin or NAD support. This can be another target area to assess for supplementation. If you see that serotonin levels are lower and the metabolites along the kynurenine pathway are elevated, then focusing on the factors that upregulate this pathway can help to shift tryptophan back towards more serotonin synthesis. There are multiple deficiencies that can impact the synthesis of serotonin. If we start from the beginning, synthesis starts with tryptophan. If there are any dietary deficiencies or even issues with digestion and absorption of amino acids, this can impair the availability of precursors required for serotonin synthesis. If tryptophan deficiency shows up on the neurotransmitter test, despite perceived adequate intake, then a gut zoomer can be an appropriate next step to determine if there is digestive insufficiency. Any micronutrient deficiencies that are required for enzymatic reactions can impair serotonin synthesis. So for example, in order to convert tryptophan to 5-HTP, the tryptophan hydroxylase enzyme requires tetrahydrobiopterin and iron. Any deficiencies there can impact that conversion. Tetrahydrobiopterin is also something that depends on adequate methylation. So impairments in methylation can also impact the synthesis of BH4. Completing a methylation panel can help to uncover whether there are issues in methylation contributing to neurotransmitter imbalances. The next reaction from 5-HTP to serotonin requires vitamin B6, which we just mentioned previously. So a deficiency here can impair that next step for serotonin synthesis. Completing a micronutrient test alongside a neurotransmitter test is extremely helpful to understand how nutrient levels may be impacting serotonin synthesis. So there's an interesting effect of vitamin D levels on serotonin metabolism. Vitamin D has been shown to activate transcription of tryptophan hydroxylase 2, which is found predominantly in the brain. This effect leads to increased serotonin in the brain. It can also act to repress tryptophan hydroxylase 1, which is found in the periphery, leading to decreased levels in the periphery. When there's a deficiency in vitamin D, however, it leads to the opposite scenario. It can decrease tryptophan hydroxylase 2 in the brain, which can lead to lower levels of serotonin in the brain. It can also increase tryptophan hydroxylase 1, which can lead to higher levels of serotonin in the blood and periphery. We're going to talk a little bit more about this when we talk about the gut. That's going to be really relevant. But for now, this effect of vitamin D really brings to light that serotonin levels can differ in the central nervous system versus the peripheral nervous system. This serotonin anomaly has been identified in conditions such as autism. Studies have shown that roughly 30% of individuals with autism show an increase in whole blood serotonin levels, so peripheral serotonin, which is likely derived almost entirely from the gut and can be an indication of dysregulation of serotonin produced in the gut. We'll talk about more about this when we talk about gut health. Animal studies showing a deficiency in tryptophan hydroxylase 2, leading to lower levels of brain serotonin, showed significant impairments in social interaction, communication, increased repetitive and compulsive behaviors, which are characteristic of autism. So you can see that particularly for this condition, vitamin D can play a big impact, and this may also be relevant for other conditions as well. This concept is also important because it focuses on the interpretation tip that we discussed, which is to match clinical symptoms with the results on the neurotransmitter test. The enzymatic activity along these pathways can greatly impact the levels of serotonin produced and other intermediates. So any genetic mutations for enzymes such as tryptophan hydroxylase, tetrahydrobiopterin, MTHFR, MAO, or even for serotonin receptors, which we're not really focusing much on in this talk today, they can all impact serotonin synthesis and activity. There are other factors that may also impact activity, such as medications and supplements. So many of the popularly recommended supplements, such as curcumin, quercetin, resveratrol, even garlic, berberine, you can see a whole list of these supplements here. This can actually slow the degradation of serotonin to form 5-HIAA because they act as an MAOA inhibitor. This mechanism has actually been one of the mechanisms that is an improved benefit from these nutraceuticals, where you look at these studies and some of these nutraceuticals have improved outcomes with mood and behavior. Part of the mechanism is due to this inhibition or slowed activity. At times, this might be a preferable outcome. Other times, it might be important to assess some of these supplements that a patient is taking to determine if it's contributing to serotonin imbalances. Also, keep in mind that other neurotransmitters can be affected by MAO inhibitors, not just serotonin. This is a beautiful visual showing the tetrahydrobiopterin cycle, called the Bioterran Cycle. Most of the, I'm going to abbreviate it as BH4 for simplicity. So most of the BH4 in our body relies on the methylation cycle, where folate is used to turn BH2, which is really down here, to BH4, which is up here. So the methylation of BH2 to BH4 provides the cofactor necessary to turn tryptophan into 5-HTP, that first rate-limiting step in serotonin synthesis. It's also required as the first enzymatic step to form catecholamines like dopamine. MTHFR genetic SNPs can reduce the Bioterran cycle and impair neurotransmitter synthesis. So this is where a methylation panel might be helpful to determine if there are any genetic factors contributing to neurotransmitter imbalances. Interestingly enough, inflammation can also lead to inhibition of the Bioterran cycle here. The serotonin reuptake transporter, which is abbreviated as SERT, is an important variable affecting available serotonin. If we review the synthesis of serotonin again, you have serotonin produced directly in the neuron. It's then stored in the vesicles by a VMAT2 transporter. Once a signal that comes down the neuron is received, the vesicles release serotonin into the synaptic cleft. That serotonin can bind to the receptors on this postsynaptic neuron or target cell. And then serotonin binds to SERT, which you can see here, to be transported back into the presynaptic neuron. And this happens because MAO is present intracellularly, and also because serotonin cannot passively diffuse back into the presynaptic neuron, it requires a transporter. A lower level of SERT activity contributes to a higher level of serotonin because less is being transported back into the neuron for degradation. Some factors that might influence SERT activity besides genetics include SAMe, methionine, estrogen, and TNF-alpha, which are known to decrease SERT activity. A high SERT activity contributes to lower serotonin because more is being transported back into the neuron for degradation. Folate, niacin, and cortisol are known to increase SERT activity. So I also wanted to discuss SERT activity because it's really important as we talk about SSRIs to really understand their mechanism and function. So while the role of serotonin in depression in the depression model is still controversial, it still remains that SSRIs are a first-line treatment for individuals diagnosed or suspected to have depression. The way that SSRIs work is by binding to SERT here so that they block the reabsorption or reuptake of serotonin back into the neurons. This makes more serotonin available for neuronal activity. One important thing to keep in mind is that while SSRIs may prevent the reabsorption of serotonin, it does not increase the amount of serotonin available or synthesized in the body. One animal study looked at treatment with 5-HTP in a slow release form in combination with SSRIs. And their conclusion was that SSRIs in combination with 5-HTP slow release could safely and effectively elevate serotonin beyond the effect of SSRIs alone. And this can represent a novel intervention for treatment-resistant depression. I wanted to include this not because it's a gold standard study, but because of the concept. Even if somebody is on an SSRI, they can still be experiencing lower levels of serotonin due to impaired synthesis. Looking at a neurotransmitter test can help understand whether this is the case. You can also see on the slide some of the SSRIs that are commonly prescribed for patients with depression. These include fluoxetine as Prozac, sertraline as Zoloft, et cetera. So these SSRIs are examples of medications that can influence serotonin. There are other medications such as tricyclic antidepressants, MAOIs, triptans, 5-HT3 antagonists, et cetera. These can all also affect serotonin synthesis and activity in the body as well. The relationship between serotonin and hormones, particularly estrogen, is important. This falls under that step of understanding the symbiosis. Estrogen has been shown to regulate serotonin activity. It does this by increasing the tryptophan hydroxylase enzyme, which is that rate-limiting step in serotonin synthesis. It can increase gene expression of serotonin receptors and the binding of serotonin with the receptors. It also inhibits and acts as an antagonist for SERT, therefore decreasing the transport of serotonin for degradation. This is the body's natural SSRI. It also has an impact to decrease MAOA activity, therefore decreasing the breakdown of serotonin. So estrogen positively influences serotonin. And when you look at symptoms of high or low serotonin and estrogen, there are many that overlap. In order to understand the symbiosis, it may be helpful to do a hormone test alongside a neurotransmitter test, which can provide important information on estrogen as well as cortisol, which remember is an inducer of that kynurenine pathway. Serotonin also has important relationships with other hormones as well. Insulin has been shown to increase serotonin. It does this by increasing the uptake of branched chain amino acids into muscles, which decreases their plasmic concentration and competition with tryptophan at the blood-brain barrier. Therefore, more tryptophan can be readily available to be transported into the brain for serotonin synthesis. So tryptophan competes with these large neutral amino acids, branched chain amino acids, leucine, isoleucine, valine, and tyrosine. So when there are higher levels of these large neutral amino acids, it can decrease that transport of tryptophan into the brain. But insulin lowers those levels of amino acids and increases that ability for tryptophan to transport. This is one of the reasons why blood sugar balance is important for balanced serotonin and other neurotransmitters as well. Due to this competition, a higher intake of branched chain amino acids or tyrosine can lead to lower levels of tryptophan transport and potentially lower levels of synthesis in the brain. Excess cortisol has been shown to have an inhibitory effect on serotonin. This is due to cortisol increasing the activity of SERT, leading to more rapid serotonin degradation. Cortisol also upregulates the kynurenine pathway, shifting it away from serotonin synthesis. Thyroid hormones can also increase serotonin levels as well. They can improve neurotransmission and receptor sensitivity. Giving 5-HTP has been shown to increase TSH levels, while serotonin depletion has been shown to decrease TSH levels. We see this in animal studies where hypothyroidism is associated with reduced serotonin synthesis. So while increases in serotonin are paralleled by increases in TSH, it really illuminates the fact that thyroid is dependent on optimal serotonin balance and vice versa. Other hormones may also have a relationship with serotonin, such as DHEA, which can increase firing activity of serotonin neurons. Serotonin is also classified as a neuromodulator, which by definition means that it regulates groups of neurons and can send messages to many neurons at the same time and can communicate with other neurotransmitters. Serotonin has interrelationships with other neurotransmitters. It has an inverse relationship with norepinephrine and dopamine. In some cases, serotonin may inhibit dopamine production, which means that low levels of serotonin can lead to an overproduction of dopamine. When serotonin is low and dopamine is overexpressed, it can lead to symptoms of high dopamine like delusional thinking, bipolar disorder, and psychosis. When serotonin is low, norepinephrine may be overexpressed, resulting in fight or flight responses and potentially leading to anxiety, panic attacks, and other symptoms of higher norepinephrine. Serotonin and catecholamines both use the hydroxylase enzyme as that rate-limiting first step. So higher activity of this enzyme along the serotonin pathway may decrease its availability for activity along the catecholamine pathway for synthesis. Oxytocin has been shown to stimulate serotonin release, and serotonin can also modulate glutamate's excitatory and GABA's inhibitory neurotransmission. This is another example of the importance of understanding the symbiosis in the nervous system, where the activity of one neurotransmitter can impact the activity of another. So we're going to focus a little bit on serotonin in the GI tract. We discussed that more than 95% of serotonin is produced by enterochromaffin cells, which are enteroendocrine cells. They make up less than 1% of the total intestinal epithelia, which is interesting considering that it produces so much serotonin. Microvilli present on the enterochromaffin cells project into the lumen of the gut. So this is the lumen, these are the enterochromaffin cells, and the microvilli here act as sensors of this luminal content, which can influence the production of endocrine signals like serotonin. Serotonin produced by the enterochromaffin cells is released into the lamina propria, leading to activation of serotonin on IPANs, which stands for intrinsic primary afferent neurons, and are present in both the submucosal and the myenteric plexus. After serotonin is released from the enterochromaffin cells, it can enter portal circulation, and it can also be taken up by platelets as they pass through enteric circulation, and that can have a big impact on blood clotting. The enteric nervous system, you can see here, contains neurons that are also capable of synthesizing serotonin as well. So there are many factors that can contribute to the release of serotonin from the enterochromaffin cells. This is not a comprehensive list, but these are definitely some interesting factors that have been identified in research. So this can occur due to vagal nerve stimulation, intraluminal distension, the presence of short chain fatty acids, ingestion, just simply ingesting a meal. It's really influenced by the presence of amino acids, hypo or hyperosmotic states or solutions, and even just the presence of glucose and galactose. Research has also found that interleukin 33, which is released in response to environmental stresses, could be a trigger for serotonin release. Interleukin 33 is an alarmin cytokine that is expressed and released from damaged tissues and during intestinal inflammation. The composition of bacteria in the gut can affect serotonin release. This includes certain dysbiotic bacteria as well as beneficial bacteria. We're going to go into some specifics on the next slide. Lipopolysaccharides can also increase serotonin release from these enterochromaffin cells. Toxins produced, such as the cholera toxin, have been shown to increase serotonin. Viruses, such as rotavirus, and in some cases, even COVID-19, especially those associated with diarrhea as a symptom, can increase serotonin production in the gut. And it's often, as I mentioned, associated with that increased motility, diarrhea, sometimes vomiting. Toxins such as BPA have also been shown to increase serotonin production in the gut. So there's really this complex interaction of the microbiota, diet, and the cells of the intestines all have an influence on serotonin production and release, and can really all be factors in altered serotonin levels seen in many GI diseases. Depending on the situation, you may need to decrease serotonin if there's an overproduction leading to nausea, vomiting, diarrhea, or you may need to increase production in situations where there's slowed motility or constipation. This slide depicts different bacterial strains and species that have been shown to induce serotonin production. Some of these may be considered more opportunistic and others more beneficial. In general, serotonin production is increased with Candida, Streptococcus, Enterococcus, Clostridium, and Escherichia. Another interesting note is that serotonin can also have its own effect on the microbiota. where specific anaerobes such as Enterococcus and E. coli can be increased as a result of higher serotonin levels. So it's really this bidirectional relationship. Researchers have found that enterochromaffin cells from germ-free mice produced approximately 60% less serotonin than those with normal bacterial colonies. But when these germ-free mice were recolonized with normal gut microbes, the serotonin levels went back up. So this is another one of those areas where patients that are dealing with GI issues really may benefit from a neurotransmitter test to understand what, even just specifically serotonin, what serotonin is doing and how it's relating to gut health. I'd like to talk a little bit about the gut-brain axis because I think this is a really interesting and important concept. I'm sure we've all heard of the gut-brain axis. It's a complex bidirectional system that exists between the central nervous system and the GI tract via the vagus nerve. Afferent vagal nerves send signals from the bottom up, from the gut to the brain. And those vagus nerve pathways might serve as a type of serotonin superhighway between the gut and the brain. This can link the emotions and cognitive centers of the brain with the gut, with what's happening in the microbiome. Vagal nerve stimulation has also been associated with increased extracellular levels of serotonin. And interestingly enough, intestinal vagal nerve activity can potentially lead to neuroinflammation, which you might see in conditions like anxiety as well. So serotonin has been shown to modulate the immune response and potentially influence intestinal inflammation. Serotonin levels can be elevated during intestinal inflammation, and serotonin can also increase inflammatory responses. Pro-inflammatory cytokine tumor necrosis factor alpha has been shown to reduce SIRT expression. One study found that there's a significant downregulation of SIRT in the epithelium of active Crohn's disease and ulcerative colitis patients compared with healthy controls. Increases in serotonin content have also been observed in experimental models of colitis, where when they were given a TPH1 enzyme inhibitor, it reduced the severity of this colitis in these animal models, just showing that relationship. Increased serotonin has also been associated with IBS diarrhea type. You can see up here, this is elevated, this is reduced. Elevated levels, they're more moderate compared to IBS diarrhea, but elevated levels can also be seen in diverticulitis and celiac disease. Celiac disease is associated with increased numbers of enterochromaffin cells, leading to higher levels of serotonin production. IBS constipation type, on the other hand, is associated with reduced serotonin, which makes sense if we think about the impact of serotonin on motility and constipation. As we discussed previously, there may be different levels of serotonin in the periphery derived from the gut versus the central nervous system. It's also entirely possible that increased peripheral production may deplete precursor availability for serotonin synthesis in the central nervous system. This may also be another factor that explains why certain GI conditions can be associated with mood and behavioral issues. We're going to talk about some intervention considerations to address serotonin abnormalities. Here are some general guidelines for low serotonin. You can use the acronym NEURO to focus on some basic recommendations. These include increasing the intake of nutrient-dense foods, exploring and then eradicating infections, using an anti-inflammatory diet, reducing stress to decrease cortisol, and optimizing digestion. There are also many non-pharmacological interventions that may help to increase serotonin levels in the body. One particular study found that self-induced changes in mood can influence serotonin synthesis, where reported levels of happiness were positively correlated with serotonin synthesis in the brain, and reported levels of sadness were negatively correlated with serotonin production. So really the interaction between serotonin and mood might be two-way, where serotonin can influence mood, but mood can also influence serotonin. There is a positive correlation between serotonin synthesis and the hours of sunlight, and this is independent of season. There are several studies that have shown exercise increases brain levels of serotonin. Exercise is associated with an increase in plasma tryptophan and a decrease in plasma levels of branched-chain amino acids, leucine, isoleucine, and valine. And since branched-chain amino acids compete with tryptophan for transport into the brain, the increase in plasma tryptophan and decrease in branched-chain amino acids can significantly increase that tryptophan availability into the brain for serotonin synthesis. Diet is another variable, which we're going to discuss on the next slide. There are three important factors to focus on when attempting to increase serotonin levels. These include increasing tryptophan-rich foods, balancing blood sugar levels, which we already discussed the important impact of insulin on serotonin levels, and as previously mentioned, focusing on anti-inflammatory foods. Increasing the intake of tryptophan-rich foods may provide increased precursor availability for serotonin synthesis. A list of high tryptophan foods can be seen in the list on the left. Specific foods such as whey protein contain alpha-lactoalbumin, which is a really rich source of tryptophan. Collagen, on the other hand, is a protein commonly used that's actually void of tryptophan. So if you have somebody that's using that daily or frequently, that might be impacting their mood or their serotonin levels. Generally speaking, you'll find tryptophan. I mean, we all, I think of turkey as like one of the most predominant sources of tryptophan, but many other animal proteins also contain higher levels, legumes, seeds, dairy, etc. There are also many foods that inherently contain serotonin. These might be banana, kiwi, strawberries, but since serotonin does not cross the blood-brain barrier, these food sources really will only have an impact on the periphery. Also keep in mind that the relative level of tryptophan compared to the other large neutral amino acids that we've talked about, like branched-chain amino acids and tyrosine, will impact serotonin. So somebody taking higher levels of those other amino acids can impair that tryptophan availability. All right, so this is a bit of a meaty slide. This is going to just describe some of the logic when we're looking at the serotonin pathway and provide information on how to determine which supplements might be appropriate depending on different scenarios. Depending on different metabolites that might be abnormal. So starting with tryptophan, if tryptophan levels are low, then increasing the intake of this amino acid is helpful. That would be a great place to start. If tryptophan is sufficient and 5-HTP is low, supporting tryptophan hydroxylase enzyme over here with nutrient cofactors would be the recommended step. And also assess to make sure that the kynurenine pathway is not upregulated, because if tryptophan levels are good and 5-HTP is low, tryptophan might be shifting more towards that kynurenine pathway. If tryptophan is low, but the kynurenine pathway is upregulated, you can consider 5-HTP supplementation to boost serotonin. You may also consider niacin or NAD support to support that kynurenine pathway with that end product of niacin production. If 5-HTP is high and serotonin is low, support this enzyme, the aromatic amino acid decarboxylase with vitamin B6. If 5-HTP is low and serotonin is low, you may benefit just from supplementing with 5-HTP. Now, if serotonin is low and 5-HIAA is high, you may need to slow this reaction here. You can look at some of those nutraceuticals that we discussed to inhibit this enzymatic activity with supplements like curcumin, quercetin, resveratrol, garlic, berberine, and vice versa. You know, if you need to upregulate this enzyme, definitely make sure that there's sufficient vitamin B2 to ensure that serotonin can be broken down to 5-HIAA. This is a really simplistic way of looking at it, but this is really following that step of understand the pathways, understand the biochemistry or the neurochemistry of the serotonin pathway, because that can really illuminate what is going on. If you have somebody with low serotonin because there is not enough B6, you know, that's really going to help you target that approach for that patient. There's some other supplements that might also be helpful in addressing low serotonin. Vitamin D3, we talked about the importance of vitamin D. It helps to transcriptionally activate vitamin D. TPH2 specifically, where it might have an opposite effect on TPH1. So ensuring sufficient vitamin D levels is important. EPA and DHA can regulate serotonin release in the presynaptic neuron. EPA also inhibits prostaglandin E2, and prostaglandin E2 inhibits serotonin. So that's part of the mechanism of omega-3 fatty acids improving serotonin. Animal studies have shown that rhodiola can increase serotonin in the hippocampus. Animal models have also shown that serotonin levels can be increased in those taking CBD. So CBD supplementation can be helpful. And then probiotics can also be helpful depending on the particular species and strain. We know that certain lactobacillus and bifidobacterium strains can increase serotonin. Definitely lactobacillus plantarum, lactobacillus helveticus, lactobacillus rhamnosus can all increase that serotonin level. And then depending on somebody's symptoms, if somebody's dealing with poor sleep and they have low serotonin levels, you can help to support nutrient cofactors to endogenously synthesize melatonin. You can also provide melatonin to just give some immediate support, because remember that serotonin is a precursor for melatonin synthesis. So most of the recommendations up until now have been focused on increasing serotonin when levels are low. However, as we've discussed, high serotonin levels can be a significant problem for many people, especially those dealing with certain gut-related disorders. So here's some recommendations to focus on when serotonin levels are elevated. The first and most important recommendation is to avoid any supplements or medications that may increase serotonin. So this can include SSRIs, or supplements containing 5-HTP. Since we know that the gut is a major site of serotonin production, assessing gut health is also an important factor. Also remember that having high levels of serotonin can lead to the predominance of certain microbes, so it's going to have its own effect on the gut. But doing a gut Zoom or stool test can provide more in-depth information about the microbes that might be contributing to these elevated serotonin levels. I would also be cautious in the situation about recommending certain probiotics that might exacerbate the problem. Serotonin levels can also be high if there are issues with degradation, which can include lower SERT and slowed MAO enzyme activity. So make sure there's enough vitamin B2, which is that nutrient cofactor for MAO. Also assess other supplements that a patient is taking, the ones that we've mentioned that might decrease MAO activity, like the curcumin, resveratrol, and quercetin. It might also be important to assess for other imbalances such as low cortisol, high estrogen, and vitamin D deficiency. While this is not a comprehensive list of action items, it's a great place to start to address high serotonin. And if serotonin is high on the neurotransmitter test, which might reflect more peripheral levels, it doesn't necessarily mean that it's high in the central nervous system. So remember to match it up with clinical symptoms for your patient. We're going to dive into our case study. This was a 56-year-old female with a history of depression, poor sleep, chronic fatigue, and constipation. So in order to assess what was going on with her neurochemistry, she completed a neurotransmitter test. She also completed some Vibrant America labs, such as HSCRP, ferritin, and vitamin D. Based on her labs, there were some significant findings that could explain many of the symptoms she was experiencing. The first thing to note was that she had significant inflammation in her body, as evidenced by an elevated HSCRP and ferritin as an acute phase reactant. Inflammation is a major inducer of the IDO enzyme, which is the rate-limiting step of the kynurenine pathway. It's also evident, based on her labs, that the kynurenine pathway is upregulated due to an elevated xanthrenic acid. Quinolinic acid was not elevated, but that's because this pathway was being shunted towards xanthrenic acid. So generally, if this was not high, the intermediates would eventually lead to quinolinic acid. But this is shifted towards xanthrenic acid, generally because of a vitamin B6 deficiency. Because it shifted, the quinolinic acid to 5-HIAA ratio was normal. So even though that can be a good indication where you're comparing the kynureninic acid to the kynureninic pathway to the serotonin pathway, if there are any shifts or abnormalities, then you might not be comparing that activity appropriately. All of the markers along the serotonin pathway were also low. 5-HTP is low, serotonin was low, and 5-HIAA was low. So supporting the intermediates along this pathway makes sense. It's also important to decrease the factors that are stealing tryptophan away from this serotonin synthesis in this pathway, which in this case is inflammation from her lab results. Vitamin D levels were also truly deficient, which can impact that serotonin synthesis in the brain. And GABA levels were also low, and this might just paint a picture for lower levels of total inhibition in the nervous system. With a low serotonin and low GABA, that can lead to lower levels of inhibition. So based on her results, we made some recommendations for supplements, which included a B complex with methyl support. This provided adequate folate for tetrahydrobioterran synthesis, B6, which showed deficient because of the increased xanthrenic acid, and also to provide some methylation support for melatonin synthesis as well. A B complex was also supportive for her symptoms because of her symptoms of chronic fatigue. And since the whole pathway was low, we also recommended 5-HTP supplementation to help immediately boost serotonin synthesis. In order to address the inflammation, she started taking fish oil at 2 grams a day and curcumin, which not only helps with inflammation, but also helps to slow MAOA activity to decrease degradation of serotonin. We talked about some lifestyle recommendations. We discussed some stress management, meditation, and this was really due to the fact that that kynurenine pathway was upregulated, and we know that excess cortisol is an inducer of that pathway. Her exercise recommendation included walking daily. This was to help increase tryptophan availability and also for sun exposure. Dietary recommendations included an anti-inflammatory diet with tryptophan-rich foods. At the 6-week follow-up, Maggie had significant improvements in energy and sleep. She noted an improvement in bowel frequency to 1 bowel movement per day, which was an improvement in her constipation. And she also reported better mood with an MSQ score decreased by 30 points. That completes the presentation, and I'm now going to open up the floor for Q&A. I'm sorry, I skipped over something actually that is important to mention. We do have some great resources available to help you with the interpretation when you order neurotransmitters. The Neurotransmitter Interpretive Guide has just been released and will be uploaded to the education portal this week. And we also offer clinical lab consults to assist with interpretation. Just make sure that when you're choosing a lab educator that they do consult on neurotransmitters. And now back to the Q&A.
Thank you, Kerri, for such an informative, relevant, and meaningful presentation. Let's now begin that Q&A.
Okay.
I did my best to answer some questions that came up while you were talking. One question was, which estrogen impacts serotonin? It's E2, right? Estradiol?
Estradiol is the one that the information in the slides is based off of, correct?
Great. And then for the amino acids that interfere with serotonin uptake in the brain, those are the branched chain amino acids, correct?
Branched chain amino acids and tyrosine.
And tyrosine, okay.
Correct.
All right. It looks like someone wanted you to repeat the slide before the non-pharmacological interventions. I don't know if you remember which slide that was.
Sure. Let's see, non-pharmacological interventions. So this slide, I hope this is what they're referring to, is the acronym NEURO. These are just general guidelines when a patient has low serotonin to try to improve and increase serotonin. So the acronym NEURO is really just nutrient-dense foods, explore infections because those infections can upregulate that kynurenine pathway and shift, it steals the tryptophan. So it steals tryptophan away from serotonin and towards kynurenine. So get rid of infections. Use an anti-inflammatory diet for the same reason, because inflammation upregulates the kynurenine pathway. Reduce stress because cortisol upregulates the kynurenine pathway. And optimize digestion. Make sure you're getting all of the essential precursors for serotonin synthesis.
That was it. They said thank you.
Okay, great.